Basically, the work that we are doing is aiming to treat retinitis pigmentosa, which is a retinal disease that causes blindness. Eventually, it starts with night vision problem and peripheral vision, and finally causes central vision loss as well. It is caused by over 60 genes that we know now, and each of these genes have many mutations, some of them hundreds of mutations. And it is inherited in different forms: autosomal recessive, X-linked, and autosomal dominant. In cases of autosomal recessive, the 2 copies of the gene are mutated and they're not functional, so there is no protein. And to treat that, all you need to do to insert a functional copy of the gene, and that produces the protein of interest. In autosomal dominant conditions, the mutated gene sometimes has a negative effect, and you have to, so you have one normal and one mutated gene, you have to actually kind of disable that mutated gene. So what we're doing, we're trying to use CRISPR-Cas9, which is a new technology that allows precise gene editing to cut that DNA and then insert a functional copy. And the method that the approach that we're taking is that we directly deliver the ribonuclear protein to the cell using nanoparticles instead of doing genomic insertion of those conditions, because this is more likely to finally result a practical way of treating this condition. It's very, very early. You know that this is only, we're doing it on cells, and then after that we have to do small animals, large animals, phase 1 human clinical trial, phase 2 clinical trial, and phase 3 until it becomes approved. Of course, I would love to see that everything goes smoothly and and we get it right the first time, but generally it's something that you have to spend do a lot of modification, improvement to make it something that's usable. The same gene therapy that is approved now in 2017, they actually cured dogs in 2001, but with that method. So look how long it took to get to human. Hopefully, because we have already a system in place, a gene therapy that has been approved, and this time will be shorter, and with the technology that is available now, but it's unfortunately still a long way to go.